I'd like to start off by introducing our next speaker, and it's my pleasure to welcome Dr. Alex Kolevzon here to speak for us. Dr. Kolevzon is a child and adolescent psychiatrist and professor of psychiatry and pediatrics at the Icahn School of Medicine at Mount Sinai. He serves as the clinical director of the Seaver Autism Center for Research and Treatment at Mount Sinai, and as the director of child and adolescent psychiatry for the Mount Sinai Health System. His research interests pertain to understanding the neurobiology of autism and related neurodevelopmental disorders, biomarker discovery, and developing novel therapeutics. Most recently, his group has focused on studying specific rare genetic forms of autism, including Phelan-McDermid syndrome, ADNP syndrome, and FOXP1 syndrome, in order to explore targets for pharmacological intervention. Dr. Kolevzon leads the experimental therapeutics program at the Seaver Autism Center, which conducts studies that range from small pilot trials in humans to multi-centered pivotal FDA studies. He also leads a national rare disease clinical research network dedicated to clinical trial readiness and studying the natural history of Phelan-McDermid syndrome. He is a frequently invited speaker regionally, nationally, and internationally, and is also extremely committed to medical student and residency education as an active teacher, mentor, and clinical supervisor. It's my pleasure to welcome Dr. Kolevzon to the stage, and thank you very much. I'm looking forward to your talk.
This is quite a stage.
Oh my goodness. So thank you very much for inviting me. I'm thrilled to be here. Great crowd. I remember we did this, this was 4 years ago, 5 years ago? Okay, everybody can hear me okay? Thank you. These are my disclosures. This is sort of what we do in academia, make sure that everyone is aware of potential conflicts of interest. I will be talking a little bit about IGF-1 today, not as it relates to FOXP1 syndrome, but Mount Sinai and the director of our center, Joe Buxbaum, have a shared patent, so that's a relevant conflict. Next slide, please. So I always start with this one because it's a happy slide. It's kids, and actually it speaks a little bit to where we started with the genetics of autism. It used to be that the best way to try to determine to what extent genetic variation accounted for autism or neurodevelopmental disorders was through twin studies, right? Where you would look at a single twin, and then if there was a identical twin that shared 100% of their genes, you'd see what the rate of autism was, and you'd compare that to a twin with what's called a fraternal or a non-identical twin that only shared half their genes, and that difference gave you some estimate of the heritability. And so in autism, going back to the 70s now, and by the way, I'm going to use autism and neurodevelopmental disorders kind of as an overarching theme, recognizing, of course, that not all of our kids with FOXP1 syndrome have autism, right? But in autism, anyway, the heritability or the percent that we think of as genetic in origin is upwards of about 90%. So it's a very kind of genetically heavy disorder. That represents a big advantage, though, in terms of trying to understand how to develop new treatments. And that's kind of what I'll try to give you a window into today. Next slide, please. So the key, going from sort of twin studies and just looking at the concordance between identical twins versus non-identical twins is technology, like everything else, right? So that the resolution and the ability to understand genes and to identify genetic changes in our DNA is just like our phones, right? Your phones have changed dramatically over the last several decades. And the same is true for genetic technology. So the resolution has gotten greater and greater and greater and greater to the point where we can see down to a single nucleotide, a single base pair in our DNA, and see if that's changed. So it used to be, if you think about it, like you're an airplane kind of hovering over New York City. If there was a building knocked out or there was like an extra building, it'd be pretty obvious. I don't want to mess with the marketing and stuff. Right, so if there's a building that's kind of knocked out, you could see it from the helicopter. Oh my God, that building's gone. Now the level of resolution is if like a tiny little pebble on a sidewalk is out of place, that you can actually see. It's so high level resolution. And then if you dig in and you think about, okay, where's that pebble located? Is that a really important street? Is that pebble going to interfere with traffic? That's kind of the level that we're thinking about now in terms of genetics. So much higher resolution. Let's go to the next slide. It's so much so that genetic testing actually is now part of the standard workup for kids with developmental delay, and certainly for kids with suspected autism. So any child that's now born, this goes back to 2013, any child that's now born with suspected autism, developmental delay, should have genetic testing. And why is that important? It's important because more and more and more cases are going to be identified with FOXP1 syndrome. So what are we up to now? 200, 300? 350, okay. So I'll give you another example with a different condition that I started working on, where when we started, it was 350. Now it's 3,500. So it's just going to be expanding and expanding and expanding because these genetic resolution, these genetic techniques are becoming more and more available, more accessible, cheaper and cheaper. The first time that I did a genetic study for a family with whole exome sequencing, which is kind of the highest resolution, I think it was $10,000 per member. So it was $30,000. Yeah, I mean, completely insane. That same test is now $1,000, which is obviously still a lot of money, but more importantly, insurance will now start to pay for it because it's recognized, it's standard of care. Next slide. So, through really huge genetic sequencing studies using enormous samples of patients with autism in this case, what was called the Autism Sequencing Consortium was actually developed by Joe Buxbaum, who's the director of our center. And what Joe decided was that by pooling resources, by bringing in literally hundreds of researchers together across, I think, 150 sites, and what's now 60,000 families, so a child that's affected and two parents, a trio, 60,000 trios, and running very high level genetic studies, we've been able to identify somewhere upwards of about 250 genes. So lots and lots of genes. And one of the genes that's sitting way at the top in terms of like level of significance, impact, level of importance is our very own FOXP1. The other ones that are some that we're working on, ADMP, ADMP and SHANK3. I won't talk about ADMP today, but SHANK3 certainly is another really important one. Each one of these genes comes with some understanding of underlying biology, and each one of these genes comes with some opportunities to develop specific targeted new treatments. So I'm going to kind of tell the story of SHANK3, or Phelan-McDermid syndrome, really as a way to kind of infuse some optimism in terms of what is going to be inevitably with FOXP1 syndrome. Next slide, please. So just a very brief primer, because I think this part's important. So, genetics, we have our DNA, the structure of our DNA. Our DNA is packaged into every cell in our body. That DNA gets transcribed to RNA. That RNA gets translated to protein. Those proteins play a critical part in our development, in our brain, in virtually every tissue in our body. Next slide. Oh, sorry, not next slide, but next, there you go. I have all these cool animations that I'm usually able to manipulate, but so it gets down to a little bit deeper, right? Where, so here's your DNA, and it's filled with all kinds of information, right? But the really critical information is occurring in these exons. But you've got promoter regions, and you've got various factors that can turn promoter regions on and off. Transcription factors, they're called. And so if you turn that promoter region off, you basically lose the translation of this DNA into a protein, and you lose the function of the protein altogether. And so there's a lot of different ways of looking at how genetic changes can impact development, but one critical way is through transcription factors. So if you can alter the DNA sequence of a transcription factor, you can change the function of that factor, and then you no longer transcribe or translate into the protein. So you're essentially missing a protein. Next slide, please. That's what's going on with FOXP1 syndrome. Lots of ways of sort of losing the function. You can knock out big sections of a chromosome. So in this case, the third chromosome, you can take literally dozens of genes and they can just get sort of wiped out. This, by the way, happens spontaneously. It happens what's called de novo. There's no way to really predict it. It's what's called stochastic or random. But you can invert it, you can duplicate it, you can insert it. All kinds of changes can lead to loss of function of these particular proteins. And these are pretty easy to pick up because they're large, what are called copy number changes. Next slide. But you can also switch even a single nucleotide, a single letter. Think about like a book. You know, if you knock out a page of a book, obviously the book is not going to make that much sense. But even if you knock out a single letter in a single word, depending on what that word is, if it's a really critical word, and it's a really important letter, you can't read the word, again, you've sort of lost the understanding of the whole book potentially. And that's what goes on with some of these single sequence variants. And so actually most of what goes on in FOXP1 syndrome, as we understand it anyway, are these very, very small sequence variants that interfere with the function of the protein. But you can also have deletions at the end, in the middle of the third chromosome that knocks out the gene also. Next slide. So FOXP1. Here we go. Third, so I don't think you need to get too tied up into sort of why is it called the forkhead box protein, but that's what it's called. It's a subfamily P. That's where the P comes from. It's kind of, you know, on the small arm of the third chromosome, so in the P region. We know that it codes for a transcription factor. Going back to what I described, transcription factors are responsible for turning on or turning off proteins. In this case, it's actually a protein that's supposed to be turning off other proteins, so it's a repressor protein. And so when you lose the function, you can kind of have too much activation, too much synthesis, which can also be a bad thing. And it's you know, what's important here is that unfortunately, FOXP1 is expressed in lots and lots of tissue. So as you know, it'll affect the gastrointestinal system, the endocrine system, but most importantly, the brain. Next slide. This is just an example of how many different ways you can have tiny little changes, tiny little changes in the DNA sequence, and it can interfere with the function of the protein. So there's a lot of stuff that can go wrong, and unfortunately, this is like a really, really critical protein. Next slide. What's interesting here is that these 60,000 cases of autism that we've now collected and done high-level genetic analyses on and identified 200 some odd genes, it seems like many of these genes, the function of them kind of converge on common pathways, of which FOXP1 is kind of within that common pathway. So the idea of the autism genes or the neurodevelopmental genes playing a really critical role in the synapse, so this is the connection between nerve cells. The idea that many of these autism proteins play a really important role in what's called chromatin remodeling, which is the way we sort of package our DNA so that it can be transcribed and translated. But here, the other major pathway are the transcription factors. So there are many, many different causes of autism and neurodevelopmental disorders that are due to sort of changes in transcription factors, where you can have either too much translation or too little translation of protein, and it disrupts function. So it's kind of fitting into this whole system now that we think is really important because, you know, the more times you can sort of develop specific treatments for one type of autism, it might have relevance to many different types of autism also. And so it's kind of nice to not have just a kind of a random protein. We're not so clear what the function is, but instead to have a protein where we know exactly what the function is, and that actually it's kind of falling in line with many other proteins that we know play really important roles in autism. Next slide. Okay, so this is kind of a conceptual framework. It used to be that we thought about neurodevelopmental disorders and we thought about, okay, there's anxiety, or there's attentional problems, or there's hyperactivity. And we would think from a very kind of top-down perspective. How do you treat anxiety? Oh, you give people, you know, Prozac. How do you treat attentional problems? You give them Ritalin, right? And it would be a very sort of symptom-focused treatment. The problem is that underneath it, all these kids had very different underlying biologies. So the effect was highly heterogeneous. And that's, you know, often the case where you can take a specific treatment and you can apply it to 10 different kids and the response can be totally different if it's just based on the symptoms. So again, top down. Shifting away, the whole field has kind of moved more towards this approach of kind of bottom up, thinking, well, let's think about from where these symptoms are derived. If this is attentional problems due to FOXP1 syndrome, it might have a particular type of treatment that's relevant. If this is attentional problems due to Fragile X syndrome, due to Rett syndrome, another transcription factor, by the way, due to ADMP syndrome, another transcription factor, by the way, all these things potentially could have their own treatments, or it could be that you find a way to kind of group them based on shared underlying biology or shared molecular pathways, and then you have a much better way of predicting what treatments might work. And so it's kind of a more targeted approach. So instead of this top-down focus only on symptoms, it's more bottom-up. Treat through the FOXP1, treat through the ADP, treat through the dermot, as opposed to just like treat the anxiety. Does that make sense? So this is the kind of framework we've been following to try to develop new treatments. And this framework is relevant for autism, it's relevant for FOXP1 syndrome, it's relevant for virtually any neurodevelopmental disorder where we can identify a gene. So it used to be we would just take autism. Now we do high-level genetic analyses, we identify the gene. We use that gene to then create what's called model systems. And you can create model systems with lots of different methods. So I used to spend a lot of time trying to convince people why a mouse was relevant to our kids, and actually it's kind of hard to imagine. But what you can do in a mouse, you can't do in a kid, and the kinds of experiments you can do in a mouse, like slicing up the brain, teach you a lot about the biology, like what's going wrong with the brain, what's happening, for example, between nerve cells, and why is learning and memory so fundamentally impaired. You can also take nowadays actually blood from kids, and you can reprogram that blood into a stem cell, and then you can induce that stem cell into a nerve cell, which is absolutely amazing. These are called induced pluripotent stem cells. So you can study a cell from a child, but a brain cell from a child in a dish, and you can understand what's going wrong with the connections, and then what happens when you apply a targeted treatment. Do you see changes in the nerve cell? Do you see changes in the shape of the cell? Do you see changes in the way that the cell can kind of conduct nerve cell signals? So really, really kind of important information that obviously you can't take from a child. But when we go through this process of identifying the gene, developing a model system, understanding the biology, and then testing a treatment in the animals, if it's promising and it seems to be safe, we can then apply that to kids, which is really a valuable kind of pathway, as opposed to just like, picking a treatment from literature that's dependent on typically developing folks and saying, oh, anxiety gets better, let's use Prozac because we know that Prozac helps keep people with anxiety. That's obviously, it could work for some kids, but it's a very kind of blunt way of doing it. Whereas this is a much more targeted way of doing it, and it's much more focused. That's not my phone. Much more focused on yeah. So does that make sense? Yeah. Next slide. I think we're going to skip over this, but this kind of goes through some of the very specifics in terms of the science that we do in the Autism Center. And it's essentially the same pathway, from the molecule to these human neurons that we can study, to the animal models, to the patients. And this is the best part. That's where the kids are. That's what we're doing with the children. That's my focus anyway. But this is a pathway that's been followed with cancer therapeutics. This is a pathway that's being followed now all over drug development. And I think it shows an enormous amount of promise also in autism and developmental disabilities. And hopefully I'll convince you of the relevance to FOXP1 in the next 20 minutes. Next slide, please. So, Phelan-McDermott syndrome. I started focusing on this condition, I think in 2008, 2009. These are kids with global developmental delay, 60 to 80% of them have autism, they have language impairments, they have attentional problems, they have anxiety, they have dysmorphic features that are nonspecific, they have MRI findings, they have sensory sensitivities. Sounds a little familiar, right? There's a huge variability across these kids also. Some of them are verbally fluent and some of them are nonverbal. So it's pretty complicated. And the same thing is true where they can have, well, let's go to the next slide. Thank you very much. Okay, so again, following this pathway, autism, identify the gene, which in this case is called SHANK3 on the 22nd chromosome. This is a really critical protein that basically serves to anchor the connection between nerve cells, and especially in what's called excitatory synapses. So excitation is this really critical process that underlies learning and memory. And this SHANK3 gene sits right there. So critical protein. If that video worked, What that video shows actually is a nerve cell that's fluorescent green in a dish. And what happens is you drop SHANK3 protein onto that nerve cell on a dish, and over the course of just 2 hours, you can actually watch as the nerve cell sprouts branches or dendrites. So what it shows is that it's actually like a really, really critical protein for nerve cell formation, nerve cell maturation, nerve cell growth. And so in this condition, Phelan-McDermid syndrome, these kids are missing one copy of their SHANK3 gene, just like our kids are missing one copy of their FOXP1 syndrome, FOXP1 gene. And in the same way, it can happen through deletions, in this case of the 22nd chromosome, or very, very small sequence variants, just of a single letter within the gene, all of which can disrupt function. Next slide. So this idea of it being kind of a synaptic gene, meaning are there deficits in the way that the nerve cells connect? This was work that was done for decades, really, including in the lab of Joe Buxbaum. So this is an experiment where they took a mice, I know, but still, they took out a copy of its SHANK3 gene, and then they sliced up its hippocampus, which is the memory center of the brain, and they looked at how nerve cells connect. And what they showed was that there was a deficit in what's called long-term potentiation. So long-term potentiation is the ability to like hold on to information, conduct nerve cell signal, and it's you see this kind of initial curve on the top. You can see it. So this is the figure on the far left. That ability to sustain the nerve cell strength or the kind of conduction signal is typical. That's a mouse that has 2 copies of its SHANK3 gene. The one below it, where it just flattens out, there's the absence of potentiation, the absence of learning. That's a mouse missing a copy, right? So it's really important. And then the figure on the right, it just shows that the morphology or the shape of the neuron is different, essentially. I spent time on this because I think that if you guys get this, it'll help you understand things in a different way and make you appreciate basic science, which is always kind of fun. And by the way, I'm a clinician. I never trained in any of this stuff. So the fact that I can understand it and make me appreciate it, you can too, for sure. Next slide. Next, there you go. So then thinking about our pathway of drug development, what kinds of medicines potentially can stimulate growth and potentially reverse this deficit in long-term potentiation. So growth factors in general are really important. We have them all over our body. One in particular is called insulin-like growth factor. And so for a whole variety of reasons, we thought about that as potentially relevant to these poor mice. And then next slide. We gave the mice insulin-like growth factor, and we actually reversed this deficit in long-term potentiation and reversed the changes in the morphology of the neuron cells. So really kind of important proof of concept that this drug is doing something meaningful in Phelan-McDermid syndrome, or at least in mice that replicate the Phelan-McDermid syndrome condition. So then what do you do next? Oh, actually, sorry, separate from that, a different group altogether out of Stanford had taken blood from kids with Phelan-McDermid syndrome, induced it into stem cells, programmed it into neurons, studied it in a dish, gave the neurons in a dish IGF-1, and saw the exact same reversal of deficits. So really exciting. We thought, okay, that's definitely worth trying in the kids. Next slide. And that's what we did, a very small, but first ever clinical trial on Phelan-McDermid syndrome with IGF-1. And we saw improvements in like totally core deficits of autism. So really, potentially disease-modifying changes because it reflected in the core deficits. So social behavior and repetitive behaviors both improved in response to this, again, very small but controlled controlled meaning that there was a placebo arm and an active treatment arm. So it was a methodologically rigorous study, but very small. So then what do you have to do after that? You have to replicate it. So we did it again with another group of patients. Next slide. And we basically replicated it to an extent. This is a lesson actually for your community, which was that we were so excited about our first study that we published it. And then the whole community knew, oh, wow, IGF-1 really works for social withdrawal in Phelan-McDermid syndrome. So then when we did another study, everybody came into it like heavily biased. And so what ended up happening was, even though we had the same general effect on the drug group, in the placebo group, everyone got better also. So we lost the effect. I always say that parents are obviously the experts in their kids, right? So I rely heavily on parents in terms of input, but parents are also the most biased in terms of expectation and hope. I mean, I'm the second most biased, by the way, but you guys are the first most biased. But this second study also gave us some really good insights into other domains that might be improving, including hyperactivity, sensory sensitivities, and actually we replicated the repetitive behavior finding, all things that are relevant to FOXP1 as well, right? So again, more evidence. Next slide. So we were really challenged with IGF-1 because it was just very hard to come by and it was super expensive. So we thought about other ways of stimulating that pathway. Growth hormone, as it happens, which is released from the brain, actually triggers the release of IGF-1 from the liver. So we thought, let's do a small study with growth hormone. Can that actually be used as a substitute because it's much more easy to access? And we did that, and we showed that actually, yes, it does increase levels of IGF-1, and it is associated with improvements. This was a very, very small, uncontrolled study, but we're now following it up with a much larger study where it's controlled, and hopefully we'll see the same thing. Next slide. Separate from the work that we're doing, completely separate, a group in China decided they should do the same thing. So they did a small but controlled study with growth hormone using the exact same paradigm that we're using now, and they actually showed improvement, not just in behavior, but also in some developmental skills, including language skills, which was exciting. And it was over a pretty brief period of time. But it's always nice when, like, you know, it's obviously, if I replicate my work, that's valuable, but if somebody else replicates our work, that's even more valuable. Next slide. And there have been now literally dozens and dozens of studies in model systems that replicate the Phelan-McDermid syndrome condition where they've reversed the changes. And all of these studies represent sort of potential drugs to think about in kids. So whether it's, you know, zinc or cannabidiol products or the microbiome, every single one of them is like, okay, maybe we should start thinking about this in kids. So just lots, like a huge flurry now. So first, high value or high likelihood autism gene, clear biology, some early clinical trials, lots of preclinical studies, lots of interest. Next slide. The most exciting of which has been our gene therapy company. So the fact that it's been 10 years and there's actually a gene therapy company that's developed what's called a mini SHANK3 gene. So they've taken out kind of some of the stuff that's not as important, created their own little mini SHANK3 gene and put that mini gene into animals. And they've seen profound changes in the animals. And they're already, even at this moment, preparing a clinical trial in kids with this gene therapy that's going to essentially replace, or the idea anyway, is to replace the function of SHANK3. So super, super exciting. Next slide. A second company is looking at a technology which is called ASO, or antisense nucleotides, where they're going to develop a molecule that attaches to the intact copy of the SHANK3 gene, because just like FOXP1 and SHANK3, it's haploinsufficiency, right? So we each have 2 copies of every chromosome. Only 1 copy is missing. It's FOXP1, or in this case, it's SHANK3. So there is an intact existing copy. So there's potentially a method to attach a molecule to that intact copy and ramp up expression to kind of compensate. So that's what the second company is doing. So these are sort of 2 gene therapies in the course of 10 years that are now being developed. And there's many more companies that they haven't declared their intent publicly, but lots of people are chasing after this. And why is that? By the way, I should have said this at the beginning. So when we started with Phelan-McDermid syndrome, it was 350 cases. Now it's 4,000. That's actually small. That's a dramatic underestimate. But why are these companies interested in these super, super rare disorders? And the reason is because it's a window into autism more broadly, right? Autism is super common. So if people see FOXP1 syndrome as opportunity to maybe start to parse this enormous area of autism, then that becomes super valuable. And so it was remarkable to me that this was happening, but that this is now happening. Next slide. So the other kind of big shift was pharma industry. It wasn't just me and our lab and a handful of our coordinators and trying to get together and bring kids in and study 10 kids with a small molecule, but actually drug companies were getting involved. So one company was Neuron. They developed an analog of IGF-1 and they started thinking about using it with Phelan-McDermid syndrome. Before that even, next slide, they had another analog called NNZ-2591, which is, again, an IGF-1-related drug that they were trying to study in Rett syndrome. And Rett syndrome, as I'm sure you're aware, right, this is an autism-related condition. It occurs almost entirely in girls. There's kind of a typical developmental course, and then they regress pretty dramatically. The gene, the MECP2 gene, is on the X chromosome, but it's a transcription factor. This drug just got approval by the FDA in Rett syndrome. So, amazing story. It's like one of the first that I'm aware of approvals for a single targeted drug in a rare developmental disorder that causes autism. Next slide. And how did it start? So this is IGF-1 again. IGF-1 in mice with Rett syndrome. Next slide. IGF-1 in kids with Rett syndrome. Next slide. 2 larger studies, controlled studies. IGF-1 in, sorry, this is not IGF-1. Now they've switched to trofinetide, which is an IGF-1 analog. Why IGF-1 to trofinetide? Because a company came in, manipulated IGF-1 and said, we have to make money off this. This is our own little commercial, patentable version of it. But it's essentially a snippet of the larger IGF-1 molecule, two randomized controlled studies, both of which with pretty clear effects. Hey, bud, you gonna help me out? Just hang out. Next slide. After the 3rd study, FDA approves it. This is just April, March, right? So this is a humongous deal. What it says is that A, the FDA cares, B, drug companies care, C, you can actually develop small molecules in rare neurodevelopmental disorders that cause autism. I guess if you went backwards, you would see the timeline, which I think goes from 2014, from the first clinical trial, to 2023. And that's 10 years ago, right? So that timeline has now gotten more condensed, and hopefully it goes faster, because I know somebody's going to ask me the timeline, and the answer is I don't know. But if this took 10 years ago, 10 years from 2014 to 2023, or 9 years, you would imagine that it's getting more and more condensed. Next slide. So the other really critical stuff, I'll tell you a little bit more about our work, because I think it's relevant to FOXP1 syndrome, and it will help you thinking about what the next steps are. Because we are essentially following in Phelan-McDermid syndrome what the Rett syndrome group did before us, which was successful, and that's exactly what FOXP1 should do in terms of following the same path, because I think it'll likely work. So one of the most important steps that we took was, after a few small trials, we decided, a few small trials are great. The Seaver Autism Center is great, but that's not going to get a registration trial. That's not going to get FDA approval. We need a whole network of expert sites. We need to prepare for what's called clinical trial readiness. So we set up a consortium. We got it funded through the NIH. It's called the Developmental Synaptopathies Consortium, of which there are a few conditions that relate to one another, but we were focused on Phelan-McDermid syndrome. And we started to comprehensively characterize what's called the natural history. So how does the condition evolve over time? That's really important for clinical trials, because if you're going to pick a symptom target, you want to make sure that it's not all over the place, right? So if it's really prominent at 2 years old, but virtually disappears by 16 years old, that's not a great symptom target from a methods perspective. We needed to validate biomarkers. We needed to understand ways of measuring things objectively, and what were sort of biological indicators that were objective of the disease condition, or being able to kind of map onto clinical symptoms. And then the third thing is we wanted to really do much better in terms of developing guidelines for clinicians in the community, right? Because when you go to your doctor and you say, my child's got FOXP1 syndrome, they turn around and they Google it, right? They've never heard about it in their life. Yeah, appreciate the laughter, yeah. I mean, even the most well-intentioned folks, and there's not a ton out there. There's not a lot of guidance. So that was important to us. Next slide. So these are our sites. We've now collected like 170 cases over the course of 6 years. We follow them out every single year. It's now a huge database. It's a biological repository. We've started to understand relationships between the genotype or the DNA and the phenotype, the clinical characteristics. So a lot of really valuable information has come out of it. Next slide. Back in 2014, I was like really ambitious and wanted to write like a review paper to try to help promote better practice parameters, because it was the same exact thing where people had no idea what to do. So they were families were diagnosed. They'd show up at their pediatrician's office, and they'd have no idea what to do. There was no kind of pathway, and so we knew very little at the time, to be honest. But we kind of did our best to summarize the literature. What do we know? And on that basis, how can we make recommendations for families and for pediatricians and for clinical geneticists to kind of just basically assess kids in an appropriate way? The Phelan-McDermid Syndrome Foundation put together this beautiful thing, which you guys could take on, I'm sure, quite easily. Carl's, he's got his, yeah. The visuals were really helpful. It's like people would actually go to their pediatrician's office and say, here, these are all the things we have to do. Next slide. And then after 10 years, literally 10 years, and a whole natural history study, we've learned a lot more. So we set about updating the guidelines. And that process started in 2019 and was iterative. We created like 12 working groups across, tons of different specialties: gynecology, endocrinology, cardiology, psychiatry. Literally every specialty group that you think could be relevant, dentistry. And we had them meet and review the literature and come up with guidelines that we thought was, kind of best practices, right? And some of it was informed specifically based on Phelan-McDermid syndrome, but some of it was just we didn't know, right? So it was informed based on developmental disorders more broadly. But now we just finally, after 4 years almost, have had our manuscript published. So it's almost 10 years after the first one, but we think we have a much better kind of working copy of clear consensus guidelines. That's going to be really, really valuable for the community. And the same thing that's happened that's relevant to your group is Phelan-McDermid syndrome has now gotten an ICD-9 code or ICD-10 code, so that when you can bill for it, essentially. So it's not like everyone has to have autism or everyone has to have something else, but you can actually have Phelan-McDermid syndrome. And when you get a genetic report that says Phelan-McDermid syndrome on it, here's the billing code, here's the diagnosis, here are the recommendations. It's very straightforward. Well, it's not very straightforward, but at least it's like more than Google will provide. Next slide. So a couple more comments just to kind of get your heads spinning when you want to dedicate some time, energy, or money. The two most important things for us, and continue to be the two most important things for us in terms of clinical trial readiness, are biomarkers and better clinical outcomes, because borrowing from the field of autism broadly doesn't work. So thinking about biomarkers, next slide. These are ways, and it doesn't matter, there's so many different kinds of biomarkers, blood peripheral markers, obviously genes are markers, DNA methylation's markers, there's brain imaging as markers. These are all markers. So we've focused a lot on electroencephalography, so EEG markers, because EEG can sort of help you understand electrical activity of the brain. It can help you get a sense of the balance between excitation, which is, as we know, is so important, and inhibition. It can help you predict treatment response. Hopefully it can help you take this huge universe of people with autism and subtype them according to different, waveforms, basically. Next slide. So Haley and Dariana take lots and lots, and Renee is here also. Hey, Renee. Take lots and lots of kids, and they put them in front of this little visual evoked potential machine, and they show them a little contrast reversing checkerboard, which is that cool thing down there. And even if you can engage for like 10 seconds, it gives you a very clear waveform. And every single one of you will have the same basic waveform that's shown on the left, right? And there's essentially two peaks. That downgoing peak reflects excitation. So you're absorbing the information from the waveform, you're stimulating your occipital cortex, and that waveform, that peak is like you kind of get it, you're absorbing it, right? And then the second peak is the kind of inhibitory response. And that kind of beautiful up and down is so characteristic of typically developing folks. And then what we wanted to see is how does it look different in Phelan-McDermid syndrome, especially because they're missing this protein that's supposed to be anchoring these excitatory synapses. And in the figure on the right, you see that that peak is essentially flat. It's missing. So over and over and over again, we can see kids with Phelan-McDermid syndrome where their excitatory peak is essentially muted. And so we're asking lots of questions like, okay, who within the universe of people with autism have a similar waveform? Maybe the same treatments that work in Phelan-McDermid syndrome can work for those kids. We're asking if we give growth factors, if we kind of stimulate growth, can we see increases in that peak? So there's a lot of cool things you can ask. Next slide. And so we did a fair amount of comparison. This P60N75 on the left is the excitatory peak, and 75 to P100 is the inhibitory trough. And what you see is people with autism, that are typically developing, siblings, and people with Phelan-McDermid syndrome all look very, very different. And the Phelan-McDermid syndrome folks are the kind of the most profoundly affected in that sense. So this is a kind of potentially a neat biomarker that we can use in clinical trials, and we are using in clinical trials. Next slide. The other thing that's really, really important in addition to biomarkers is just better ways to measure change. So you guys have all taken questionnaires that look at like attention and anxiety and sensory, and they're irritating, I'm sure, and maybe many of the questions have nothing to do whatsoever with your kids, and they feel kind of irrelevant, and they're very frustrating, and they frankly don't necessarily capture symptoms that well, because they're not specific to FOXP1 syndrome. They're just not. They were developed for intellectual disability broadly, they were developed for autism broadly, and so a significant chunk of the questions are essentially irrelevant. In the case of Phelan-McDermid syndrome, most of these kids are minimally verbal or have no words at all. And so lots of the questions of these questionnaires are like, oh, how does your child express this? Parents hate that. It's just, So next slide. In the Angelman syndrome community, in the Rett syndrome community, and slowly now in the Phelan-McDermid syndrome community, there's been a lot of focus on trying to develop more kind of refined versions of existing tools, but are specific to the characteristics of their particular syndrome. And I think that's a really valuable approach. And that's something you can start to do when you have more natural history data. And that's something that's been done here. And in fact, the tool that was used in that Rett syndrome trial was the approvable tool by the FDA. So these are now considered to be like valid and specific to the phenotype. Next slide. It's 1:15. Oh my goodness. So this is stuff that you guys already know. Here we've come to FOXP1 syndrome. Here's some of your cute kids, some of whom are now much bigger. Oh my goodness. I saw one of them outside. Barely recognized him. Next slide. Lots of sort of initial efforts. An early paper written by Ray Lozano when he was in San Diego. Now he's at Mount Sinai. I think it was one case. So this is what year? I can't see. 2015? 2015, yeah. Next slide. We did a little bit more work. I think this is a couple years later. I'm starting to accumulate more data about what the clinical characteristics look like, more cases. Next slide. Started to understand, oh, these kids are kind of unique, right? There's a few things that are going on there that are not specific necessarily, but much more common. This is so important because when you do a clinical trial, you have to pick symptoms that are common across all the kids. You can't pick some sort of unique small symptom. I mean, seizures is a great symptom to potentially target, right? Because it's so objective and easily measurable. But if it's 10, 15% of the kids, that's a much different trial than like ADHD or compulsive behaviors or collecting stuff, right? Next slide. And this was our kind of attempt, my 2021, sort of based on minimal amounts of data, at least put something out there that says, hey, this is what you should do when you get the diagnosis. Next slide. And here's what we did, right? I mean, you guys all know this, I'm sure. This is very rudimentary, but at least it's like something of a framework. And I think it, hopefully provides some guidance and some reassurance that there are steps that one should take across all these different domains. I'll say one comment about autism, right? I think people have mixed feelings about this because autism is different in so many ways. I think some kids with FOXP1 syndrome definitely, they meet criteria and they clearly have autism and parents kind of relate to that. Others don't, but still want that diagnosis. That's actually fine because that diagnosis is a pathway, it's actionable, it's prescriptive in a way, and it helps get access to services. So I would feel comfortable with that. Next slide. We're going to summarize. So your work, my work, our work together, hopefully will be to do more to understand the natural history. So I think we're doing now adult studies. So we want to kind of follow kids over time, see how things change. We definitely need to be very careful about understanding the clinical features, how they're unique, how they're different, what are common across kids, how do they change over time. We absolutely need to develop biomarkers, and every time we see kids with FOXP1 syndrome, we're putting them through different kinds of biomarker studies, whether it's eye tracking or visual evoked potentials. For you guys who have been through our center, you know how much we've put you through. Laughter, appreciate that. And we need better clinical outcome assessments. So that could mean developing a new tool that's specific to FOXP1 syndrome. It could mean taking an existing tool and refining it a little bit. It could mean taking 10 tools and pulling pieces from it, what's called a composite measure. We could just break down raw items and decide which among these items are relevant to FOXP1 syndrome. I'm 4 minutes over, but I'm happy to stop. I'm good. Questions?
So I would like to thank you so much, Alex, on behalf of the foundation. It's a little token. Yeah, you're done for the presentation. We're going to do questions in the breakout group.
Okay.
We'll go right into the breakout groups, but there's a little token for thank you for appreciation for it. You really got the FOXP1 Foundation started with Carl many years ago. We wouldn't be here if it wasn't for your contribution and for Seaver Center contribution. I know it's a lifeline for so many of us. And your information just shows you how much we have to do. You were just at the very beginning. And as parents ask so many questions, it's like we're the first generation of this. So we have to contribute to science so that we can get somewhere in 20 years from now. And you give us a roadmap. Thank you. Thank you very much.